Hi everyone, my name is Edel O'Toole. I'm a professor and academic dermatologist at the Blizzard Institute at Queen Mary University of London. I'm delighted to welcome you all to today's ESDR Kitchen live webinar. So the ESDR introduced these during the pandemic so we could have interactive sessions with our members and people throughout the world who are interested in skin biology. And today we have a freshly baked session where we discuss two recent exciting papers, one from Piotr in Science and the second from Hemanth in the American Journal of Human Genetics. I'm going to pass you on to Curdin Conrad, who will introduce the first speaker.
Thanks, Edel. So I'll make it short so we can move forward to the first presentation. I have the pleasure to introduce Piotr Konieczny. Hope that was more or less properly pronounced, Piotr. He's a postdoc in a Bernard B. Levine postdoctoral fellow in the immunology department of the New York Grossman School of Medicine in New York in Dr. Shruti Naik's lab. So really an outstanding lab, really hot right now with amazing publications, and Piotr will present one of these amazing publications that just came out in Science a few months ago. So first of all, congratulations on that one, Piotr. Just very briefly on Piotr, he had his PhD in Poland and joined then Shruti's lab in 2019, where he focuses on the crosstalk between immune cells and epithelial cells, both in wound healing and cancer. He received actually several awards, and one very important one was also the Research Award for Young Researcher from the ESDR. There are some others as well, so we're really looking forward to have you here, and just the stage is yours. Thanks, Piotr.
Thank you so much. I will share my screen. Thank you so much for the invitation and introduction. So I will briefly talk today about our recent published story, how interleukin-17 signaling drive proper tissue repair. As you know, skin houses different types of the immune cells, and a few years ago, Rachel Clark published the beautiful paper where she found that billions of T cells and many other lymphocytes localized in healthy skin around her follicle, and these cells rapidly respond to microbiomes, nutrition, noxious agents, or carcinogens challenge, what we call right now homeostatic immunity or homeostatic lymphocytes. So we were very interested how this extrinsic microenvironment signal derived from the lymphocytes, from the immune cells, co-opt with the primitive cellular stress response during skin repair and during tissue repair. In particular, we were interested how these homeostatic lymphocytes drive signal direct tissue repair, and what's the migratory program downstream of this interaction between lymphocytes and skin epithelium during skin repair. So first, we'll average single cell sequencing and site-seq sequencing to define the lymphocytes landscape early after wounding. So we use full thickness sponge biopsy, and we isolate lymphocytes from healthy control, from healthy skin, as well from the wound's edge of day 3 and day 5 post-wounded skin. And we found that not only regulatory cells, but also effectory cells are rapidly remodeled during tissue repair. As well, we found that type 17, the cells involved in type 17 immunity, are highly proliferate and expand early after wounding. So as you know, probably our gamma T transcription factor is the key transcription factor which controls type 17 immunity. This transcription factor is expressed by different subset of the immune cells and control the function of this immune cells by regulating the expression of pro-inflammatory cytokine, including interleukin-17A, F, and interleukin-22. So we decided to focus on the small population of the cells, and we try to understand what's the role of the cells in tissue repair. So we decide to knock down this lymphocytes in mice. We use GFP knock-in mice. So basically, we have the GFP is in the locus of gamma T transcription factors. So basically, we have the mice with non-functional lymphocytes which are not able to express pro-inflammatory cytokine. We punch them and we analyze the kinetic of the wound closure as well as the length of the migrating tongue. And we found that mice lacking gamma T lymphocytes heal significantly slower, as well they have the shorter migrating tongue compared to wild-type mice. So these results suggest that the cells somehow are involved in tissue repair, skin repair. So then we did a lot of work to figure out which subset of gamma T lymphocytes are main driver in our phenotype, and we found that our gamma T, gamma delta T cells are the main driver responsible for our phenotype. We also found that the main mediators between lymphocytes and damaged epithelium is interleukin 17A. We inject intradermally interleukin 17A into the wound's edge of our gamma T deficient mice. So basically, we try to rescue our mice and we analyzed the length of the migrating tongue in these mice, and we found that after injection, the migrating tongue was significantly longer compared to PBS injected mice. We also found that interleukin 17 RC receptor was the main receptor responsible for IL-17 signaling, where we knocked down this receptor specifically in keratinocytes, and we analyzed and we found that the migrating tongue in these mice was significantly shorter, and these mice healed significantly slower compared to wild-type mice. So far, these results suggest that maybe IL-17 is involved in tissue repair. So the big question was, how does IL-17 signaling promote tissue repair and rehabilitation? So we decided to look much more broad. We, in the tissue context, we apply special transcriptomic techniques, which allows us to detect the gene expression in the tissue context, and we compare the wound's edge of wild-type mice as well with wound edge of GFP knock-in mice, which is marked here on the red. And we found that wild-type wound's edge highly expressed the genes which are involved in IL-17 signaling pathway, which was not surprise for us. But we found that the genes which are involved in HIF-1 alpha pathway are highly expressed in the wild-type wound's edge compared to GFP knock-in wound's edge, which can suggest that maybe IL-17 is involved in HIF-1 alpha activation. So to validate, we look on the expression of and the localization of HIF-1 alpha in the migrating tongue of wild-type mice as well or gamma T deficient lymphocytes, and we found that GFP knock-in mice at significantly lower number of nuclear HIF-1 alpha in migrating tongue, as well IL-17 RC deficient mice mimic this phenotype. We also confirm, of course, the role of that HIF-1 alpha is important for skin repair. We knock down HIF-1 alpha in keratinocytes, and we found that mice lacking HIF-1 alpha in keratinocytes heal significantly slower compared to wild-type mice as well. They have the shorter migrating tongue compared to wild-type migrating tongue. So as you know, HIF-1 alpha is in normoxic condition is rapidly degraded by proteasome, but in hypoxic condition, it's stabilized, translocated to nucleus, where drive the expression of HIF-1 alpha dependent genes. So we were start wondering, okay, HIF-1 alpha is driven by hypoxia. So what's the level of the hypoxia between wild-type wound edge as well or gamma T deficient wound edge? So we use two different techniques, pimidazole. So pimidazole is the molecules which you inject into the mice, is metabolized, and based on the level of the hypoxia, you can detect the hypoxia localization using immunofluorescence staining. As well, we use hydroxy probe, so the probe which directly measure the oxygen level in the tissue. We insert the probe into the wound edge and we measure the oxygen. And in both cases, we found that the hypoxia level between wild-type wound edge and GFP knock-in wound edge is pretty similar. So which can suggest that IL-17 maybe drive the HIF-1 alpha expression activation independent on the oxygen sensing. So to verify this, we moved to in vitro study. We established organoids culture derived from epidermal stem cells, and we culture our organoids with interleukin-17, and we found that indeed that interleukin-17 was sufficient to directly regulate, activate HIF-1 alpha in our organoids culture. As well, this activation was dependent on IL-17RC receptor. So IL-17 signaling IL-17RC receptor and HIF-1 alpha is quite far from each other. So we did a lot of work to try to understand the molecular mechanism which is involved in this activation. And from our preliminary data, as well from already published data, what is established in the field, we know that one of the pathway which is involved in HIF-1 alpha activation is mTOR. So we decided to block mTOR using rapamycin in our organoids culture, and we found that indeed mTOR pathway was crucial for HIF-1 alpha activation after IL-17 stimulation. I will not go to the details, but we also found the upstream molecular pathway which was involved in HIF-1 alpha activation, which involved IL-17RC receptor, AKT, mTOR. So this molecular pathway was crucial for HIF-1 alpha activation after IL-17 stimulation in our organoids culture. And of course, the downstream of this molecular pathway is metabolism, is glycolysis. We found that IL-17 activate glycolysis in our organoids culture, and as well we found that glucose transporter 1, which is involved in glycolysis, is significantly lower expressed in in the migrating tank of gamma T deficient mice, as well IL-17RC and HIF-1 alpha deficient mice. We block glycolysis using 2DG inhibitor, both in vitro and in vivo, and we found that mice after 2DG treatment had significantly shorter migrating tank, as well the keratinocytes migrate significantly slower after glycolysis block, which can suggest that IL-17 dependent glycolysis is crucial for proper tissue repair and keratinocytes migration. So just summarize what we found. We found that small population of lymphocytes, which we call our gamma T, gamma delta T cells, actively proliferate. So there are not circulating cells, there are resident cells. They highly express interleukin-17, which activate epithelial cells in the migrating tank of the wood's edge via interleukin-17RC receptor to activate the whole molecular pathway, including AKT and mTOR, which are necessary for optimal HIF-1 alpha activation, which is translocated to nucleus, where drive the expression of glycolysis genes, which are necessary for proper skin repair. We also found that chronic hypoxia, long-term hypoxia can block mTOR. That's why we need the additional signal provided by IL-17 signaling for optimal HIF-1 alpha activation. So in the end, I would like to thank all my lab members, especially Shruti, for her support. This work is done with collaboration with UX Inc., or amazing bioinformatician, my collaborators from NYU, as well Karolinska Institute, my funding source, and you for your attention. I will be happy to take any question. Thank you so much.
Thanks, Piotr. The alarm just went off in our building, actually, so Hemanth had to move. But I'll start the introduction to him, and then I'll probably have to get out of my office. So Hemanth completed his PhD and postdoctoral training in human genetics and DNA repair in Dundee, Scotland. Subsequently, he moved to the Blizzard Institute, Queen Mary University of London, to work with Professor Indrajit Dokal and functionally characterize several novel genes and pathways that become problematic in human hematopoiesis. He is now a lecturer in genetics and molecular biology, and his research program continues to investigate bone marrow failure syndromes. Dyskeratosis congenital, of course, is a disorder with a skin phenotype. Hemanth is going to talk about his recent paper in the American Journal of Human Genetics, where he had an interview as a highlighted author. And you can read about his interest in cricket and fancy cooking, as well as this fascinating paper. So thank you, Hemanth.
Thanks very much for this invite. I had to run away from a fire alarm in the building and get myself into a calm corridor. So you might listen to some noise. I apologize for that. So I'm going to talk about my recent paper, which as Edel introduced. Before going into that, I'm just going to give a brief introduction into the disease and the biology that I've been working for the last decade. It is called Dyskeratosis Congenital, and in short, it's called DC. It's characterized by a triad of features such as nail dystrophy, skin pigmentation, and oral leukoplakia. The life-threatening illness is they develop bone marrow failure, and some patients often get cancer, such as MDS or acute myeloid leukemia. DC is also considered as a telomere biology disorder, and I'll tell you why. Telomeres are the end base pair repeats of 6 base pairs at the end of our chromosomes, and they're maintained by this enzyme called telomerase. It's a complex of this protein, telomerase reverse transcriptase, and a RNA molecule, TERC. Mutations in either that affect the function of these proteins, and which in turn affect the maturation and stability of telomerase enzyme, or the trafficking, or the telomere itself, the proteins that involved in protection of the telomeres, such as the Shelton complex proteins, or telomere replication. All these defects in all these functions have shown to cause short telomeres in DC individuals. If you look at this metaphase spread of a normal individual, you can see the telomeres are as highlighted in yellow. They'll be glowing, whereas in DC, you see some chromosomes do lack this signal. And if you measure the telomere length in DC cases, they're obviously very short when you compare to the control individuals. So at Royal London here, we have accrued around 500 to 600 index cases of DC. And the genetics of DC is very variable. It could be either X-linked, which means only males are affected, or either autosomal dominant, where the disease features are present in the individuals where the mutation is present, or it could be incomplete penetrant as well, where you have a mutation, but the disease is occurring only in certain individuals and family members. And it could be autosomal recessive, where if the mutation is present in homozygous or compound heterozygous pattern, then only you see the disease features. But when in only heterozygous fashion, you don't see it. Some of the sporadic cases have no family history either. So how our story started? In the last 2 years, we have compiled a list of telomere length regulators that have been published in various studies. And these regulators are excellent collection of telomere length regulators for our common population. And we compiled a list of these telomere length regulators and compared them to exome sequence data in 189 DC patients. So we identified a series of 8 independent families with heterozygous variants in the gene TIMS. And if you look at the cases from these families, they are so homogeneous in their phenotype presentation. And you can see that they have developed skin pigmentation, nail dystrophy in almost all of the cases. So what is TIMS? TIMS is a thymidylate synthase gene that is predominantly involved in the nucleotide metabolism pathway, and especially in the synthesis of DTTP. It is involved in the conversion of DUMP to DTMP, which is deoxyuracil monophosphate to thymidine monophosphate. So based on this, we measured the DUMP and DTMP levels, and we found out that the patients are accumulating DUMP levels and their conversion rate is actually lacking or is actually reduced in the patient cells. So what is this accumulated DUMP doing? So we looked at the telomeric specific sequence, digested the patient's DNA, and with a specific enzyme called uracil DNA glycosylase enzyme UNG, and we did a PCR for the telomeric signal, and we observed that there is a reduction of the telomeric signal. We also seen that the telomere length in these cases is reduced, and in turn we also have noticed that they have increased DNA damage response in terms of the gamma S2X staining. So what's fascinating about this study was actually when we encountered this problem, which I'm going to tell you now. The cases have the mutation, have the disease features, but the mom or the dad who is having the variant does not have the disease features. So this actually told us, is it something autosomal dominant and incomplete penetrance that we've seen before? Or is it something that will be completely missed? Or is it just a random coincidence? So what we did measure is the TIMS level itself at RNA level. And what we all identified was, in the cases that RNA is reduced by 50%, and so does in the parent carrier, as well as in the wild-type parent. This is quite striking. And when we looked at the protein level was reduced almost 90%, but we don't see that reduction in the parent or the wild type. And at the same time, we have looked at the sensitivity of these patients to a specific drug called 5-fluorouracil. And this 5-fluorouracil is actually a direct target of the TIMS molecule. And we found only the patient cells have hypersensitivity to this molecule. What we also identified was the TIMS level in patient actually fluctuate over the passages, as so does in the control as well. So there is some sort of evidence that the TIMS is actually getting translated for the remaining 50% of transcript we are seeing. So we labeled this RNA, we cross-linked this RNA, and then what we have done is we pulled the ribosomes by antibody HSP70, which binds to the mature polysomes. It's a chaperone of the polysomes, and we subsequently subject to RT-PCR. And strikingly, what we found out that the wild type RNA, which is actually made in the cells, it's not actually binding to the ribosomes, which in turn incited that there is some kind of a transcriptional, post-transcriptional factor that is inhibiting its binding. And this is another experiment which we did, where we have actually showed that when we transduced wild-type TIMS RNA into the patient cells, we tend to rescue the endogenous TIMS RNA. Endogenous, and this is also dose dependent. When we transduce at the reduced MOI, it tends to even rescue them. So which is suggesting that there is some kind of a competition that's happening, and there is a factor that is competing endogenously with this molecule in order to inhibit the TIMS expression that is expressed in the 50% wild type which is expressed. So over our studies, we realized that the TIMS regulation is actually very complex. TIMS neighboring gene called enosF1 is actually transcribed in the opposite direction, and both TIMS and enosF1 share the 3' UTR. And also, there are variants that are shown in the TIMS and enosF1 have been shown to be the predictors of the fluoropyrimidine-related toxicity. And what I mean is, like, when cancer patients are given any fluoropyrimidine drugs, they tend to have severe toxicity effects such as hand and foot syndrome, as you can see in this picture. So we looked at that and we took that information, we sequenced the whole locus of this in the genome and where the parent's DNA is available. And what we identified is in the other parent, which does not have the TIMS variant, the variant is in green, it does have a haplotype that is indeed involved in the lower expression of the TIMS. So when you have a combination of this haplotype coming through, and we also identified in few other families, patients, in the cases from few other families, there are intronic variants in this locus. So there is a combination effect of the variants that are burdening this locus that is coming into the case itself, and that's how it's hampering TIMS expression. But at the moment, it's just a hypothesis at this point. So we measured the enosF1 levels, and we found out that the patient's cells have higher enosF1 levels. And what you see is over the passages, when compared to the control, these levels seems to increase. They're always higher the eNOS-F1 when you compare to the TIMS level. So we thought, okay, if we do an RNA inhibition of eNOS-F1, so what's going to happen from here? So bingo, this is a clincher experiment where we actually knock down the RNA by RNAi, the eNOS-F1, and that rescued the TIMS expression. And we'd also seen a bit of modest rescue in the toxicity to the 5-FU, which indirectly is saying to us that there is a TIMS and eNOS-F1 is what causing the severe TIMS deficiency, what you've seen in this patient. So in summary, what I'm showing here is the parent with a variant in the TIMS and a variant in eNOS-F1. And this high eNOS-F1 post-transcriptionally silences the TIMS, causing low severe TIMS deficiency in the patient, and which in turn alters the nucleotide metabolism and causing telomere length attrition, increasing predisposition to the premature aging features and genome instability in these patients. And this is a classic example, I would say, that where the insights from Achieva studies has been taken in identifying a Mendelian traits for a disease phenotype. And I think this is, we think this is the first evidence of linking the nucleotide metabolism to the telomere maintenance in the human Mendelian disease. And we believe that this digenic inheritance of this variants from 2 different genes in the neighboring units and operating as a functional unit, this kind of mechanism could be applied for other Mendelian disorders too. However, the bigger picture is actually the establishing the TIMS eNOS-F1 ratio in 5-FU related cancer therapies. So if you have high TIMS eNOS-F1 ratio, then I presume that these patients would be developing severe toxicity, hand-foot syndrome. On that note, I would like to end my talk by thanking the families and the clinicians and my lab members and the funding bodies and the collaborations that have participated in this study. And thank you very much. I'm sorry again for all this palaver. So thank you.
Thank you very much, Hemanth. I think that was probably the presentation I've ever witnessed in the most difficult situation or circumstances while being evacuated from your office. I hope everything's fine, but you managed and really, really well. Thanks a lot, despite all these disturbances. There are no questions so far, so please, if you have any questions to Piotr, Hemanth, please put them into the chat function and we'll hand them over to the 2 speakers, which gave 2 excellent presentations. So Piotr, just maybe to start with a very practical or maybe simple question. I mean, if you say that 17A interferes with or drives wound healing, but when we do blockade of 17A and F or both of them, you usually do not see a delay of wound healing. How do you explain that?
I think it depends on the, like, you mean using the antibodies, right?
Yeah.
So I think that with the antibodies, the problem, like, you need to test it how efficient the system works. We block very specifically IL-17RC receptor, which is very specific for IL-17A, right? We have IL-17RA, which is much broad for the IL-17 family, but RC is very specific for RC. For IL-17A, and we see delayed wound healing. We also did IL-17RA full knockout. We also see delayed in the wound healing. So I think it's more about like technical how the antibody works and if it's efficient to really block the full IL-17 in the tissue context. Because like there are also other evidence that IL-17 drive the tissue repair, right? And also in the skin. So another thing is, I think like we need to consider it is the microbiome. Between each animal facility, microbiome is different. And this can also interfere with IL-17 response.
And you think that in the human system, the whole thing, is relatable, that it's the same mechanism that relates to the human system?
So we see in humans that the cells are expand in the wound, we tested the human wound sample. So we see that the cells expand. We see that HIF1 alpha is activated in the wound edge. We stimulate human organoids in vitro with IL-17, and we see that they migrate faster. As well, we block HIF1 alpha in human keratinocytes. We block glycolysis, and like we see similar effect on the human keratinocytes, primary keratinocytes.
And could not be that there's more, redundancy in the human system, because if you block, for example, 22, you also get wound healing, which is completely abrogated. While if you block 17, it's not so much, so that in the human system you have more redundancies. I
think like it's like everything about the balance. The same is with IL-17. When we think about, like most people think about IL-17, that is a pro-inflammatory bad cytokine, right? Induce the psoriasis. But I think it's not necessary. It's more about like how amount IL-17 which tissue cells require for homeostasis. We did a lot of work to try to find the balance between IL-17, and we see if you give mice more IL-17, you can block migration and wound healing. Like the cells not migrate, and you have like expansion of you more mimic psoriasis than migration. So I think it's everything about the balance, and like of course in the humans, I think it's the system is more efficient and everything have to work. So I think it's just the balance. Just maybe a last question to you from my side, so we give Hemanth a little bit of time to dislocate. I'm not sure where he's going, if he's evacuating finally the building, because you also lost Edel. She never came back. So I hope everything's fine with you there. Do you think it could be that it's depending on, it showed on hypoxia, that in inflammation you have most of the time a lot of oxygen and a lot of hyperemia, and that in a wound healing where the wound is atonic and has a hypoxia, as you showed, that then it becomes more important for wound healing, the 17 and your glycolysis part, or is this independent? You mean independent on the hypoxia? Yeah, so it's just if you have hypoxia, it's still necessary, right? It's still dry. I think like you need this additional signal for this optimal activation, because hypoxia also change. You have very hypoxic microenvironment on the beginning of the wound, but when the wound starting closing, the hypoxia also change. So I think that's why we need the additional signal, which is provided by immune cells, which also are, when we look on the kinetic of the immune cells, it's very dynamic, right? So that's why I think like it's not only hypoxia and not only IL-17, it's just they are co-op together to drive the proper tissue repair. Yeah, thanks a lot. We still don't have any questions, so maybe switch to Hemanth, and still don't have Edel. Thanks again also for you, talk. Great. One question: do you think this system that you found is this relatable directly to other genetic diseases which have the same traits? Did you have some candidates already?
Well, I certainly believe that, because now finding a disease locus and you have this neighboring genes that are transcribed in the opposite directions, and if they're sharing a common 3' UTR, so they must kind of have this kind of inhibitory mechanisms against each other as the short hairpins form post-transcriptionally. So if you have a variant effect in either of these genes, then one should be negating an effect from on the other, or one should be causing an effect on the other. So I certainly believe that this could be something like a look forward thing in terms of the disease locuses, where you see these neighboring genes. There are digenic diseases that have been depicted before, but it has to be something like a mutation in one locus and the other mutation in the other locus, and then they somehow in 3D conformation bind against each other, or post-transcriptionally make their own inhibitory RNAs against each other, or one is having a gain of function in terms of protein as a transcription factor binding towards the other. Such as there are few cases of retinitis pigmentosa, which is one form of blindness, and the other form of a blindness with the deaf disorder, the Bardet-Biedl syndrome. There has also been cases of showing the digenic inheritance.
And is there a link to consanguinity? So that, you know, because you need the two genes, that you have one gene in from one patient or more frequently in one family tree and then the other one coming in, or is this completely independent?
I think it has to be as a digenic, you know, one in one, you know. So as I showed that there is a haplotype that is coming from one of the parents along with the TIMS variant that is, you know, in working with in trans with the other. So I guess the inhibitory effect has to work as a functional unit, both coming together. So it has to be both rather than one just coming from one. If it is only coming from one, you see that in the parent, they had the TIMS variant, but they don't have any disease features. And we've been going back and forth with these parents, asking like, do you have disease when you were growing up? You know, like any disease features that came and diminished? And they said like, absolutely not. You know, so it has been quite a big study for over the time, because we were confused as well. You know, why this effect, what we are seeing, is only in the kid rather than in the parents, you know, who has the variant.
What do you expect the kids of these kids have? Because if there doesn't seem to be like a trait that seems to be, you know, is it a new mutation, or is this just pure luck and it will be gone, or any idea?
That is a very interesting question. Obviously, that the kids of these kids, okay, over this locus, if you sequence it, and you tend to identify some variants and some combination. So I kind of feel that it is a variant burden that is coming onto the locus along with the exonic TIMS variant that is acting in cis. So yeah, you know, as with all the genetic diseases, it's a matter of 1 in 4 or 1 in 2, or yeah. So the chance, you know, so.
I was just curious because none of the parents had it. I mean, by chance, you would expect that at least some would also have it. Yeah. But we'll see next generation.
Hopefully, yeah.
Okay, no questions coming in. I'm not if everyone was evacuating in the meantime, also from the chat. So we'll cut it short here. Thanks a lot for both of you for 2 excellent presentations, particularly also for Hemanth under these conditions. The chats will be online in a few days, as usual, on YouTube on our channel. The next ESCR Kitchen will be in mid-November, mid-September, on hair research on different in vitro and ex vivo models. There's some hot topics on hair research lately on how to grow again hair. So this will certainly push that research again. Then just a reminder for the late breaking abstracts, deadline is next week. So if you have any really late breaking abstracts and late breaking research, please submit them. And then obviously we hope to see you in a month in Amsterdam for the annual meeting. It's going to be an amazing meeting, an amazing venue, and I hope to see you all there. And again, thanks a lot for joining in, the 2 of you, for giving the talks. Edel, I hope she's fine. She never came back, but I assume everything's okay. And see you soon next week or next time. Thanks a lot. Bye-bye.
Thank you so much. Bye-bye.
Thanks, Piotr.